Welcome to the Deep Dive. Our mission, well, it's pretty simple. Cut through the complex research, get you the key insights straight up.
Exactly. And today we're wading into some really important territory, aquatic biodiversity, and invasive species.
We're looking at a paper that asks a pretty pointed question, actually.
It does indeed. It's titled, "What Are We Protecting?" On the Silent Arrival of Anodonta cygnea to Spain. Lead author is Omar Sánchez and colleagues, with correspondence from Arias.
And this deep dive is really about a fundamental issue in conservation, isn't it? If you don't know what you're looking at.
You can't protect it. Simple as that. And the creature here is the freshwater mussel, part of the Unionoidae group.
Right, and these aren't just any shellfish. You've called them ecological heavyweights. Why?
Because they absolutely are. They're keystone species. Think of them as the river's natural filtration system.
Okay, like living water purifiers.
Precisely. They cycle nutrients, clean up pollutants, stabilize habitats. They're vital. When they disappear, the whole system can, well, collapse.
And the problem is they are disappearing globally. Pollution, habitat loss.
And invasive species, which is exactly where this paper comes in. In the Iberian Peninsula, you've got a native mussel, Anodonta anatina. It's struggling.
And then there's this other one, Anodonta cygnea. Its status has been debated. Maybe it's non-native, maybe not.
And the kicker is they look virtually identical based on the shell alone. It's incredibly difficult to tell them apart. Decades of confusion.
It's a morphological trap.
That's the perfect term for it. And why this paper is so important, it uses DNA, molecular evidence, to confirm a large, thriving population of A. cygnea.
The potentially non-native one.
Yes, found in Catalonia, Spain, where it wasn't really thought to be established like this. A silent arrival indeed.
Okay, let's dig into how they discovered this. What was the situation before this finding? The native mussels were already in trouble.
Definitely. The whole Iberian Peninsula is seeing declines. You've got species like Potamida littoralis struggling, and huge efforts going into saving critically endangered ones like Pseudonio auricularis. The ecosystem is stressed.
And sometimes stress reveals things. What was the trigger here?
It was the drought, a really severe one across northeast Iberia, Catalonia in 2022 and 2023.
I remember seeing reports, reservoir levels just plummeting.
They dropped incredibly low, down to 10, maybe 15% capacity in some places. An ecological nightmare, obviously.
But it exposed the lake beds, the shorelines.
Exactly. And that's when researchers walking the shores of the Darnius Bodella Reservoir found them. Not just a few, but really high numbers of anodonta mussels. Adults, juveniles. Suggesting healthy reproducing populations.
Okay, wait. If the native A. anatina is known to be vanishing there, finding any mussel thriving in large numbers is weird.
It's more than weird. It's a huge red flag. It immediately suggested this might not be the native species everyone assumed was struggling. It contradicted the narrative.
So the objective became clear, because shell out what these mussels actually are.
Precisely. They realized they had to go beyond just looking at the shells. They needed genetic analysis.
For these populations in Spain, but also, you mentioned a comparison site.
Yes. Crucial comparison. They looked at a site in nearby France as well. The goal was to nail down the species identity and then figure out, okay, what does this mean for managing these ecosystems?
It's often these extreme events, like droughts, that suddenly pull back the curtain on changes that have been happening silently for years.
Absolutely.
Yeah.
Systems under pressure often reveal hidden invaders, species that might actually cope better with disturbance than the natives do.
So how did they go about confirming the identity? If looking at them doesn't work, it's all down to the lab work.
It really is. They focus on two key locations. The Spanish site, that Darnius Bodella Reservoir in Catalonia.
Okay.
And the French site, Lac de Villeneuve de l'Avajo. Importantly, it's only about 30 kilometers away, just across the border.
Why that specific French lake?
Well, it also had lots of mussels, but its ecology was known to be pretty degraded. It's full of the invasive Asian clam, Corbicula fluminea, for instance. So similar mussel density close by, but potentially a different story.
That close proximity really sharpens the question, doesn't it? Two spots nearby, mussels look the same. But are they?
Exactly. And to answer that, they turned to DNA barcoding. They sequenced a specific gene fragment, mitochondrial cytochrome C-oxidase subunit I, usually called COI.
Ah, the COI gene. That's like the universal barcode for animals, right?
For much. It's a standard marker used worldwide for species identification. Very reliable for distinguishing even closely related species like these.
So they got the DNA sequences. What next? How do you analyze that?
Standard procedure, mostly. They used Sanger sequencing to get the DNA code. Then they compared these sequences against huge online databases, GenBank, using a tool called BLAST, basically asking, what does this DNA match?
But they went further than just matching, didn't they?
Oh, yeah.
Here-
To understand the evolutionary story, the relationships between these mussels and others in Europe, they did phylogenetic analysis, building a kind of genetic family tree using maximum likelihood methods.
Okay.
And really importantly, they did haplotype network analysis. They used software called PawPart for that.
Haplotype network analysis. Sounds complex. What does that tell you that the basic ID doesn't?
Think of it like tracing genetic lineages across geography. A haplotype is a specific version of that COI gene sequence.
Right.
If the mussels in Spain all share a haplotype that's super common, say in Central Europe, but hasn't really been seen in Spain before.
It suggests they didn't originate here recently.
Exactly. It points strongly towards a recent introduction, maybe hitching a ride from somewhere else, rather than being some long-lost native group that just reappeared. It helps trace the origin story, the movement.
So this level of genetic detail was absolutely necessary because the shells were just misleading.
Completely misleading. Couldn't rely on them at all.
Okay, the moment of truth then. After all that DNA work, what did they find in the Spanish reservoir, Darnius bodella?
The results were unambiguous. Every sequence they got from that reservoir, it was Anodonta cygnea.
Wow. The potentially non-native one.
Yes. The match was near perfect, 99 to 100% identity with known A. cygnea sequences in the databases. And crucially, not a single specimen of the native A. anatina was found there. Not one.
That's pretty definitive. So what about the French site just 30 kilometers away? The one that looked the same?
That population at Lac de Villeneuve-l'Arajo confirmed as Anodonta anatina, the native species.
Incredible. So side by side, essentially you have two visually identical populations, but one is native, one is likely introduced.
And that perfectly illustrates the paper's main point. You cannot rely on morphology alone for these mussels. It's simply not reliable for conservation or monitoring. DNA is essential.
And the novelty here is significant. This is the first time A. cygnea has been confirmed using DNA in this specific river basin, the Muga.
First time in the Muga Basin, yes. And only the second DNA-confirmed population in all of Catalonia. It proves the silent rival wasn't just suspected, it has happened and established.
And did the haplotype analysis back up the recent introduction idea?
It did. The A. cygnea found in Spain all shared one single haplotype.
Just one type.
Yep. And it turns out this specific haplotype is very common across the rest of Europe. It's not unique to Iberia.
So it's like finding a suitcase with a label from Berlin in your backyard in Barcelona. It probably didn't originate there.
That's a good analogy. It strongly supports the hypothesis that these Spanish mussels are likely descendants of recent arrivals from Central European populations, not some ancient hidden native group.
Okay, so the arrival is confirmed, the genetics point to introduction. What does this actually mean for the ecosystem there? What are the consequences?
Well, it raises some serious concerns. A. cygnea is known to be a pretty robust competitor.
So it could push out the native species.
That's the major risk. Competition with the already struggling native A. anatina. It could also potentially alter the habitat in ways that further disadvantage native species. Freshwater ecosystems are fragile.
And this reservoir, Darnius bodella, it wasn't exactly a pristine environment to begin with, was it? The paper mentions it's known for other invasions.
Oh, absolutely. It's cited as a known invasion hotspot. It's where scientists first recorded several non-native fish in the Iberian Peninsula. Things like common bream, asp, roach, pike perch.
Fish from other parts of Europe.
Exactly. And even the invasive spiny-cheeked crayfish showed up there first for Iberia.
Oh.
The paper notes its fish community actually looks more like what you'd find in France than in the rest of Spain.
Which leads to the pathway question. If this is a recent jump, how did A. cygnea get there? Mussels don't exactly walk across borders.
No, they don't. The authors point a strong finger at sport fishing.
Anglers. How?
Well, mussels have a parasitic larval stage called glochidia, tiny little things. They need to attach to the gills or fins of a fish host to develop and disperse.
Ah, so the fish are the taxis.
Precisely. And places like the Muga River and the nearby Ebro are really popular destinations for anglers from all over Europe.
So maybe anglers brought in boats or gear or even bait fish carrying these non-native fish hosts?
That's the most likely scenario. Unintentional transport. And remember those exotic fish already established in the reservoir, the bream, the roach.
Yeah.
They are known hosts for A. cygnea glochidia. So the earlier fish invasions actually created the perfect conditions for the mussel invasion to follow. The taxi service was already running. so to speak.
Wow, it's like a cascade effect. One invasion enables the next.
It often works that way. Plus, the paper suggests that the decline of native mussels might have created empty niches. Ecological space was available.
Like an empty apartment in a busy city.
Kind of. The native tenants moved out, or their numbers dwindled, leaving resources and space available. And A. cygnea seems quite good at colonizing these modified habitats, like reservoirs, so it moved right in and thrived.
This brings us back to that conservation paradox. What's the immediate takeaway for people managing these rivers?
It's critical. Any conservation program in that area targeting Anodonta mussels needs an urgent review, if they're just going by visual ID.
Well, they might be spending money and effort protecting the invader.
Exactly. They could be unwittingly helping the non-native A. cygnea while thinking they're saving the native A. datina. It's a potential major misallocation of precious conservation resources, a huge mistake.
So the message is clear. Integrate molecular tools. DNA analysis isn't a luxury anymore, it's fundamental.
It has to be baseline for effective conservation, especially with cryptic species like these.
What about the future? What needs to happen next, research-wise?
The authors are clear. More sampling, more sequencing. They need to map the full extent of A. cygneas spread across the Iberian Peninsula.
See how far it's gotten.
Yes, and monitor existing populations. Are they displacing A. annatina completely? Are they hybridizing? We need a much bigger picture. This study opened the door, but there's a lot more to understand.
So the big takeaway from this deep dive, it seems to be that protecting biodiversity effectively absolutely starts with knowing what you're protecting.
Fundamentally, yes.
And this silent arrival of A. cygnea, helped along by human activity and maybe amplified by drought. It just underscores how complex managing invasive species is, especially as environments change.
It really does. It forces us to constantly reevaluate, to
Yeah.
use the best tools available, and to be aware that the ecological landscape under the water might be shifting faster than we realize. You can't just rely on how things used to look.
A sobering but crucial lesson.
Indeed.
Thank you for joining us for this deep dive.